DNA replication model. In this video, we will present our science fair project for biology subject. The scientists from 11S1 participating in these works are Angélica Ponce, Hilary González, Josué Valdés, Crislin Rodríguez, Sofía López, and Valeria Citón. All living organisms contain DNA as part of their genetic code. Replication of these special molecules needs to occur because existing cells divided to produce new cells. Each cells require a full instruction manual to operate properly. When cells in your body need to repair or replace dead cells, replication takes place. Big question. Do you want to know how cells replicate DNA? Objective. The objectives of these projects are to model and describe DNA replication process. Materials: acrylic paint, wooden sticks, foamy, small and medium-sized styrofoam pellets, large styrofoam sheet, manila paper, crazy glue, hot silicone, scissors, cutter, brush, malleable foamy, hot silicone gun, and pipe cleaner.
Procedure: replication process. First, we paste the manila paper over the lace styrofoam sheet. Then we cut and paint the wooden stick with 4 different colors. Each stick should have 2 colors. In this case, we match blue with green for half of the stick, and the other half is painted red with orange. For doing the model of replication, we have to mold the malleable foam in order to make the different enzymes, proteins, and part of replication, and let them dry. You will have to make some of the enzymes that participate in the replication on the top of the model when it is done, for more precision and ease. After that, we cut foamy strips and paste them together by the ends in order to form 2 large strips. Then we start to paste everything together with hot silicone. First, we paste the sticks in different parts over the strip of foam in order to make the double helix form of the DNA. We bend the strip. We also paste the fronds made with the malleable foaming in the place where they should be depending on the function that they develop in DNA replication. In order to raise the model, we use large wooden sticks and paste them with hot silicone. We reinforce it, putting malleable foaming in the base of each stick.
Cell and chromosome. Shape the malleable foaming in an X form to make the chromosome and a circular form to make the nucleus and let it dry. Cut part of the center of a styrofoam palette and then paint the palette with blue color. Then paste pieces of pipe cleaner on the styrofoam pellet in different places. Paste the nucleus in the center of the styrofoam pellet. Use a wooden stick to link the chromosome with the cell. Paste a rolled piece of pipe cleaner in the lower right end of the chromosome. Join the pipe cleaner to the starting end of the foamy strips using hot silicone. DNA model. Paint the small and medium-sized styrofoam pellets with two different colors, blue and red. We also paint wooden sticks with the same pattern as the wooden sticks of the replication model, blue with green and red with orange, then let them dry. Then we link the wooden sticks with one medium-sized styrofoam pellet on each of its sides. To put everything together, we use a large wooden stick in the middle and we paste the wooden sticks linked with the medium-sized styrofoam pellets onto the large stick. In order to separate the wooden sticks, we use small styrofoam pellets between each medium pellet.
Deoxyribonucleic acid is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and viruses. The two DNA strands are composed of simpler units of monomers called nucleotides. Each nucleotide has three parts: a phosphate group, a ring-shaped sugar called deoxyribose, and a nitrogen-containing base. One molecule of human DNA contains billions of nucleotides, but there are only 4 of them: adenine, thymine, guanine, and cytosine. They only differ in their nitrogen-containing bases that are thymine and cytosine, called pyrimidines because they have a single ring structure. Also, adenine and guanine called purines because they have a double ring structure. Thymine always pairs with adenine and cytosine always pairs with guanine. This is known as the base complementary rule because the DNA strands are complementary to each other. This means that one strand can serve as a template to the other strand.
The DNA nucleotides of a single strand are joined together by covalent bonds that connect the sugar of one nucleotide to the phosphate of the next nucleotide. They create the backbone of this molecule. The DNA double helix is held together by hydrogen bonds between the bases that are stacked in the interior in pairs. The two strands of the helix run in opposite directions so that the fifth carbon end of one strand faces the third carbon end of its matching strand. The carbon ends refer to the 5-carbon structure of the deoxyribose sugar. This antiparallel orientation is important to DNA replication. As you just heard, DNA is a complex molecule which contains the genetic information of living things. The process by which this molecule is copied is called replication. In eukaryotic cells, it takes place during the cell cycle in the synthesis stage, specifically inside the nucleus. There, the 46 chromosomes of your DNA are replicated once in each round of the cell cycle. As a result, replication assures that every cell has a complete set of identical genetic information.
Steps of replication. DNA itself does nothing more than store information. Enzymes and other proteins do the actual work of replication. Many proteins are enzymes that function as catalysts. They decrease the activation energy and increase the rate of chemical reactions. Replication process starts when enzymes begin to unzip the double helix at numerous places along the chromosomes, called regions of replication. At each division in eukaryotes, there are many replication of regions from which DNA synthesis proceeds bidirectionally.
Firstly, topoisomerase enzyme relaxes the DNA strands. Secondly, helicase unwinds the double helix, that is, hydrogen bonds connecting base pairs are broken. This separation of the two DNA single strands creates a Y shape called a replication fork. Third, single-strand binding proteins attach to nitrogen bases and prevent the two strands from joining. An enzyme called primase creates an RNA primer which establishes a 3' beginning that will be the starting point for synthesis of the new strand. Next, DNA polymerase synthesizes the new strands complementary to each template. DNA polymerase only adds nitrogen bases in one direction, from 5' to 3'.
Also, polymerase proofreads and corrects newly made nucleotide incorporation errors.
On one template, replication occurs in a smooth, continuous way in one direction. This strand is called the leading strand. It goes from 5' to 3', so replication here is uninterrupted. On the other template, the lagging strand cannot be made in a continuously way because it runs in the opposite direction from 3' to 5'. So the DNA polymerase can only make this strand in a discontinuous piece-by-piece way. This series of small chunks are called Okazaki fragments. Each fragment is started with an RNA primer. The DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction called Okazaki fragment. The next RNA primer is added further down the lagging strand. Another Okazaki fragment is made and the process is repeated again.
Once new DNA has been made, the enzyme exonuclease removes all the RNA primers from both new strands of DNA. Another polymerase then fills in the gaps that are left behind. Finally, other enzyme called DNA ligase is in charge of connecting the fragments of DNA to each template in both strands. The result is two complete and identical molecules of DNA that are exactly like the original double strand, each new DNA molecule with one strand from the original molecule and one new strand.
How is the process of DNA replication? DNA replication is called semi-conservative because one old strand is conserved and one new strand is made. Replication is fast and accurate. In every living thing, DNA replication happens over and over again, and it happens remarkably fast, especially in eukaryotes. This process proceeds from hundreds of origins of replication along the chromosome, so the process takes just a few hours. In prokaryotes, as their DNA is circular, there is only one origin of replication.
On average, around one mistake is made for every 10 billion nucleotides that are replicated. The process includes over a dozen different types of enzymes and other proteins to run correctly, and the cell has multiple mechanisms to ensure the accuracy of the DNA replication. For example, DNA polymerase and isomerase enzyme have a proofreading function to correct errors.
DNA replication is a very important process for any living things in order to produce new cells, replacing the dead ones, and they should have the exact same information as the original one in order to avoid any new mutations. DNA replication is a process that should be done very carefully. Many proteins, enzymes, and others are responsible of making sure that this process is developing. Also, they all have a specific function in replication. This process also ensures that the characteristics of an individual will be passed down to generations because the DNA contains the genes and traits of every living thing.
While doing this project, we learned a lot of things. Some of them are that in the replication process, a lot of enzymes, proteins, and others are needed because they develop a specific function in the replication in order to separate the DNA or put it together or develop a lot of other functions. We also learned that this process ensures that the next generation will have some of the traits and characteristics of this individual, because the DNA is the one that carries all the genes that will be passed to the next generation. We also learned that the DNA replication is one of the most important processes for any living thing. Why? Because the DNA replication helps to make a new cell that will replace a dead cell in that organism. If it is not done correctly, then this information will lead to a mutation and it will affect the whole individual.